Hello everyone, what's up? Hope you all are doing well. So in today's video tutorial I am going to talk about ELISA. ELISA, that is a biochemical technique used to diagnose the presence of particular antigen or antibody in the sample of the patient. So ELISA stands for enzyme linked immunosorbent assay. That means it includes enzyme activity of enzyme. Immunosorbent means you need to absorb some sort of immunological antigen or antibody present in the sample of the patient. So let's start with ELISA. ELISA is an analytical biochemical assay, and it uses enzyme for the detection of the substrate, for the detection of the results. So what are the requirements to perform ELISA? You need microtiter plate. This microtiter plate contains large of numbers of wells. You fill your sample, you load your sample in these wells for the diagnosis of particular antigen or antibody in the patient sample. This microtiter plate is made up of polypropylene plastic material. The next, you need pipette. This is also known as the micropipette because it is used to pick up the microliters of the sample. Next, you need transfer pipette for the transfer of the sample. Next, this is how your microplate looks like when it gives results. The results will be colorful. So one more thing, let's suppose why we perform ELISA. Let's suppose a person has been diagnosed with HIV infection. Now you want to clarify, you want to justify whether the person is really suffering with HIV or no. If the person will be suffering with HIV, then the virus will be present in the body of the person, and you can perform ELISA to identify the presence of particular antigen in the sample. If the person's sample contain HIV virus, then it will give colorful results in this microtiter plate. But if the person is free of infection, then it will not give any sort of coloration, means the person would not be identified with HIV virus. So let's see how this all happens. First of all, there are three types of ELISA: direct ELISA, sandwich ELISA, and competitive ELISA. In this video, we'll only talk about direct ELISA and sandwich ELISA. About competitive ELISA, we'll discuss about it in next video. So first of all, the direct ELISA. When we talk about something direct, something straightforward. So in this process, what you will do, you will take the microtiter plate, then you will take the sample of the patient that you want to diagnose whether it is containing any antigen or no. So if the sample of patient is containing antigen, then obviously the antigen will stick to the surface of the microtiter plate. Then after loading the sample of the patient, you will wash the microtiter plate with particular reagent, and after washing, what will happen, excess of the antigen that is stuck on the walls, on the surface of the wells will be washed out. Then the next step will be you will use the labeled antibodies that will be labeled with particular enzyme for the further results. Now if your sample is containing particular antigen, then it's gonna to stick on the walls of the well, and if the antigen is present on the walls of the well, it will be recognized by the specific antibody. That means there would occur the interactions of antigen antibody. Then the next step is, now you want to see whether there has occurred any interaction between antigen or antibody. To check this system, to check this process, you will add a specific substrate for that particular enzyme. If the complex has been formed, okay, one more thing, after interaction between antigen and antibody, you will once again perform the washing step. Like if the antigen is available on the walls of the well, the antibody will make interaction with the antigen. But if the walls of the well have not any kind of antigen over there, then antibody will not remain there, antibody will be washed out by the rinsing process. Okay, then now if the antibody is available, then that means enzyme is also available over there, and if the enzyme is available, there will occur a reaction between substrate and enzyme. There will occur the production of color that will give your positive results, like the person is really suffering with the infection of HIV. So this is the direct ELISA, means directly we are using specific antibody regarding a specific antigen. So it uses basically antigen antibody interactions, and one more thing, it eliminates the possibility of cross reactivity. Cross reactivity means when your particular antibody react with any other antigen that might be present in the sample and you don't want, that possibility will be eliminated by this sort of ELISA. The next ELISA is the yummy ELISA, means it's the sandwich ELISA. So we all love sandwich. Well, in sandwich ELISA we make a sandwich like structure in the wells of the microtiter plate. Basically antigen is sandwiched between the two antibodies. Let's suppose these slices are the two antibodies, and these cheese and these vegetables are the antigens that has been sandwiched between these two antibodies. So in case of sandwich ELISA, it's used for detection of antigen in patient sample. Once again we want to detect the presence of antigen, so it's a quite bit more accurate method than your direct ELISA. So let's see how to perform it. The first step is the antibody. Instead of that antigen, you will coat the well by using non-antibody, okay, on the microtiter plate. The next step will be wells are washed to remove the unbound antibodies. Third step is you will add the sample of the patient that you want to know whether it containing particular antigen or no. If it does contain, then obviously that antigen will interact with that particular antibody. The next fourth is you will add a primary antibody molecule. So this green one is the primary antibody molecule, so it will interact with antigen. If the patient sample is containing antigen, then obviously it will make interaction with antigen. The fifth step is you will add the secondary labeled antibody. That secondary labeled antibody will target the primary antibody. If the wells are containing primary antibody, obviously secondary labeled antibody will combine with it. And make sure after each and every step you are performing the washing step. Okay, then the next is after addition of each antibody, washing step will be performed, as I have already told about it. So why? Because it will remove the excess or unbound antibody that are present in the sample, and it will also avoid the error. So let's see once again, this is the microtiter plate. You have coated antibody, non-antibody, then patient sample. If it contain antigen, obviously it will interact with its specific antibody. Okay, now if the antigen is present, primary antibody will definitely interact with it. If primary antibody is present, obviously secondary antibody will bind with it. Then the last step is addition of substrate for detection. You will add a particular substrate for that particular enzyme. If the whole process has been occurred, then the enzyme would be there, and if the enzyme is there, obviously you are gonna to get the particular color, you are gonna to get that particular color as a result. If a particular color doesn't appear, that means the patient is not suffering with that particular disorder. Thank you.